response, which would be T cells. And T cells are going to provide defense against intracellular antigens, cells infected with viruses or bacteria, and unfortunately transplanted cells as well. There are a number of different kinds of T cells, and while they are going to provide a defense against intracellular antigens, they're also involved in helping the B cells get to the different kinds there. So they're involved in controlling the immune response as well. Some T cells kill directly, others are going to release chemicals that help regulate the immune response. So they are a little more complex, actually they're much more complex than B cells in classification and functions. There are two major populations of T cells that are based on cell differentiation antigen on their surface, a protein, and it's a glycoprotein, it's got sugars on the surface of the T cells. And those two basic types of T cells are CD4 positive T cells and CD8 positive T cells. And that is going to relate to their functional abilities as well. So the first one, cluster or cell differentiation, CD4 positive cells usually are going to mature into T helper cells. Those T helper cells are going to secrete things that increase the activity of B cells, get them going really well. They can work on other T cells, they work on macrophages, they are kind of directing an adaptive immune response. And some of those T cells will become memory T cells and provide a memory response the next time you see it. And some of them will differentiate to become regulatory T cells, which help moderate immune responses, turn them down when it's necessary. The other population is the CD8 positive T cells, and those are generally cytotoxic T cells. They are the T cells that are able to find cells that are infected with things like viruses and kill them directly. Again, they also have some that will become memory T cells. There is also a population called regulatory T cells. And if we know what they are doing, if they're activated, then we can define them as helper, cytotoxic, or regulatory. If we don't know what their antigen is, if they haven't seen an antigen yet, we refer to them as CD4 positive or CD8 positive T cells. So the major types of T cells, immature lymphocyte leaves the bone marrow, goes to the thymus, matures to either express a T cell receptor and the CD4 molecule, or a T cell receptor and the CD8 molecule. When those naive T cells leave the thymus and begin their jobs of surveying the body for antigens, upon activation, the CD4 positives become T helper cells or regulatory T cells, while the CD8 positive T cells become cytotoxic T cells, the effector cell. T cells only respond to fragments of antigens, and those fragments of antigens have to be displayed on the surface of cells within a major histocompatibility complex, or as we talked about before, MHC proteins. So T cells have to see something foreign, but they have to see it in the context of something that is self. They don't just see antigenic determinants the same way that antibodies do. And in order for that to happen, we need antigen-presenting cells, and antigen-presenting cells are vital to change naive T cells into effector T cells. So those MHC proteins come in two different classes. Class 1 MHC proteins are displayed on almost all cells. Red blood cells do not have class 1 proteins on them, but almost all cells have MHC class 1 proteins. Those are the signs that say, I belong to this organism. This is the one that without that class 1, that natural killer cells would go after the cell. The other type of MHC class 2 proteins, are displayed only by the antigen-presenting cells. So Cells like dendritic cells, macrophages, and B cells express MHC class II. Uh, both MHC class I and MHC class II are synthesized in endoplasmic reticulum. They pick up peptide fragments. Where they pick up those fragments is a little different, but they pick up peptide fragments and put them along with the receptor onto the surface of the cell. So MHC class I proteins are on the surface of every cell, and within the binding site of an MHC class I, there is a small peptide. Same is true for MHC class II, except they are only found on antigen-presenting cells. So the MHC class I proteins bind with little short fragments, 8 to 9 amino acids, and they get those amino acids from things that are built within the cell itself. So they are called endogenous antigens. They're being built within the cell. So an endogenous antigen could be a self-antigen. Most of them, many of them, are going to be the pieces of proteins that are being made at the same time that the MHC is being made by the cell itself. But if that cell is infected or is abnormal for some reason, some of those fragments are going to be from a virus that's infecting the cell, an intercellular bacteria, whatever it is, being built inside the cell, picked up by MHC class I and expressed on the surface of the cell. And again, class I proteins are on almost all cells. Class I MHC is crucial for CD8 cell activation. It acts as an antigen holder. It's got a part that is self, the MHC itself, that the T cell recognizes, and it's got the piece of something else, the non-self, to inform a cytotoxic T cell that there's a microorganism hiding in that cell. Class II proteins pick up larger fragments, longer fragments. They go from 14 to 17 amino acids, and they get those amino acids from exogenous or extracellular locations. So these antigen-presenting cells are phagocytizing things, and the things they're phagocytizing and breaking down, parts of those get put into MHC class II proteins. And MHC class II proteins are recognized by CD4 positive cells, so T helper cells, and they signal that, again, self, the MHC class II is self, but foreign, the 14 to 17 amino acids in the midst of that MHC, activate helper T cells. So class I proteins, all nucleated cells, almost all nucleated cells. Class II, antigen-presenting cells. Class I, CD8. Class II, CD4. Class I, endogenous. Class II, exogenous. So that gives us what we call MHC restriction. CD4 and CD8 have different requirements for MHC proteins that present them. CD4 positive cells that become T helper cells are restricted to binding only class II MHC proteins, and those are found on antigen-presenting cell surfaces. On the other hand, CD8 positive cells that are going to become cytotoxic T cells are binding to class I proteins, and those class I proteins are found on almost all cell surfaces. That would include antigen-presenting cells. Once they're activated, cytotoxic T cells will seek out the same antigen on class I MHC proteins on any cell and kill that cell to prevent viral infections. It's a dilemma for how antigen-presenting cells are allow CD8 positive cells to become activated by non-endogenous phagocytized proteins. Normally we get endogenous proteins presented on MHC1. But dendritic cells have ways to get around it. They can engulf dying virus-infected cells. They can have connections directly with other cells so that they pick up infected proteins. There are a few ways around it. So T cells can be activated only when antigen is presented to them, either in MHC1 or MHC2. And that activation is at least a two-step process. There has to be antigen binding and there has to be at least one co-stimulation. In fact, there are numerous co-stimulation, co-binding things that happen between antigen-presenting cells and T cells to make sure it happens correctly. But we'll focus on the simplest example. They're going to occur on the same antigen-presenting cell, and they are going to be required for clonal selection of the T cell. So antigen binding. T cell antigen receptor has to bind to an antigen MHC complex on an antigen-presenting cell surface, which means that T cell has to perform a double recognition. It has to recognize the self MHC and the foreign antigen it displays. So we say it's displaying that antigen in the context of self MHC. Binding that TCR complex is going to trigger intercellular signaling pathways that begin the process of T cell activation. But other surface mark proteins are going to be involved in completing that activation. CD4 and CD8 are vital to help maintain coupling during antigen recognition. And then co-stimulation. The complete T cell activation requires the T cell to bind one or more co-stimulatory signals. If you don't get that co-stimulation, process called anergy can occur, and that would make the T cell become tolerant to that particular antigen, and they would be unable to divide, and they would not secrete their cytokines. But it's a very important process. It's a two-step process. It's a safeguard against accidentally getting T cells working against our own cells. T cells that are activated would then enlarge, proliferate, response to cytokines. They differentiate and perform functions according to their class. Primary T cell responses peak within about a week, similar timeline to what we had with B cells producing antibodies. Apoptosis occurs between 7 and 30 days. The activated T cells are hazardous because they have large amount of inflammatory cytokines, and inflammation can get out of control. That could really result in hyperplasia, and memory T cells are going to stay around and mediate a secondary response similar to what we saw in B cells. So clonal selection involves, again, recognizing non-self in the context of self, so dual recognition. Antigen presentation on dendritic cells engulfs that dendritic cell engulfs an exogenous antigen, processes it, displays it in the context of MHC class 2. That CD4 positive T cell recognizes that antigenic determinant, or what we'll call an epitope, in the MHC class 2. CD4 strengthens that bond. Other co-stimulatory molecules come along and activate the T cell, and that T cell clone proliferates. We get clonal expansion. This process involves a lot of cytokines, and cytokines are chemical messengers of the immune system. They're going to help with cell development, differentiation, responses within the immune system. A couple groups of them include interferons and interleukins. I just looked up on PubMed. The highest number interleukin I could find was interleukin 38. So there are a huge number of interleukins, and when I left the research field, they were coming out every few days with new interleukins. But we know some of them are more important in, or we say they're more important in T cell differentiation. Interleukin 1 is a very important one, released by macrophages, stimulates T cells to release another interleukin called interleukin 2, and to make more interleukin 2 receptors. Interleukin 2 is a very important growth factor. It acts on the same T cells that release it. It acts on other T cells. It encourages activated T cells to divide more rapidly. There are other cytokines that help to amplify and regulate both the innate and the adaptive responses. Gamma interferon helps make macrophages angry macrophages, so they're even more effective. So like I said, there are tons of interleukins and some interferons out there. Interferon alpha and beta have antiviral effects. They activate natural killer cells. We talked about them earlier. Gamma interferon or immune interferon is secreted by lymphocytes, works on T helper cells, works on macrophages. Interleukins 1 and 2 we talked about. Interleukin 4 tends to promote T helper cells that switch antibody production to IgE. Interleukin 5, I'm not real familiar with, but here it says it works on mast cells, activating eosinophils. Interleukin 10, secreted by macrophages, it inhibits macrophages and dendritic cells, tends to turn down the innate immune response. 12 is another important one, secreted by dendritic cells and macrophages, stimulating cytotoxic T cells and natural T cells. And interleukin 17, another one that I'm not real familiar with, is important in the innate and adaptive immunity in the recruitment of neutrophils. There are also suppressor factors, which is kind of a generic term for cytokines that suppress the immune response. Transforming growth factor beta, TGF beta, is a suppressor factor similar to IL-10. Tumor necrosis factor, TNFs, produced by lymphocytes and macrophages. They promote inflammation, enhance phagocyte chemotaxis, and non-specific killing. And a lot of the drugs you see advertised for things like arthritis, psoriasis on TV, those are monoclonal antibodies that interfere with tumor necrosis factor. All right, let's take a look at the specific effector T cells. The first one most first discovered, earliest, best understood, are the helper T cells. They play a central role in adaptive immune responses. They help to activate both the humoral and the cellular arms of an immune response. They're primed by antigen-presenting cells, and they become helper T cells. They activate B cells and other T cells, cause proliferation in T and B, and they secrete cytokines that recruit other immune cells. Without T helper cells, we do not get a good immune response. And unfortunately, we know this because of the human immunodeficiency virus, which specifically targets T helper cells and eliminates CD4 positive cells, and that hence eliminates immunity. T helper cells activate B cells, they stimulate them to divide, produce more antibody. Responses can occur without T helper cells, but they tend to be short-lived. They tend to be IgM only, and they're a limited number, and they are known as T cell independent antigens. Most antigens are T cell dependent antigens and require T helper cell co-stimulation. CD8 positive T cells require T helper cells to become activated and become the cytotoxic T cells that they are meant to be. They can cause dendritic cells to express co-stimulatory molecules that are required for CD8 cell activation. Effector T cells can also amplify innate defenses. They activate macrophages, making them more angry, potent killers, mobilize lymphocytes and macrophages, and attract other types of white blood cells. There are also subsets of T helper cells. This research has been going on and starting to be better defined. T helper 1 cells, most aspects of cellular immunity, these are the common ones. T helper 2 cells tend to shift things towards the kind of defenses you need for parasitic worms. That would include eosinophils, IgE antibody responses, humoral immunity responses, and that because of the IgE nature of a lot of allergy-based antibodies, they probably promotes allergies as well. T helper 17 links the adaptive and innate immunity by releasing IL-17, and we think that may have a role in regulating autoimmunity. Cytotoxic T cells directly attack and kill other cells. The activated cytotoxic T cells are circulating in the blood, moving through the lymph and the lymphoid organs, through the body, looking for their antigen. Once they find a virus-infected cell, a cell with an intercellular bacteria or other parasite, sometimes cancer cells if they've changed enough, or transplanted cells, which we would get in transfusions or transplants, they would take over and attack them. They do that by delivering a lethal hit. That lethal hit involves the release of proteins called perforins, which poke holes in membranes, and granzymes, which get into the cell and activate apoptosis in the target cell. And cytotoxic T cells, they bind specific membrane receptors on the target cell and can stimulate apoptosis externally as well. Cytotoxic T cells are similar to natural killer cells. Natural killer cells recognize abnormalities. Cytotoxic T cells recognize abnormalities. Natural killer cells are more specific for those that lack MHC class I protein. So they're not seeing an antigen in the context of self, they're seeing a lack of self on the target. They will also target cells that have antibodies on them, and they also target proteins that are expressed when a cell is under stress. They use the same mechanisms as cytotoxic T cells. So together with cytotoxic T cells, natural killer cells are an important part of immune surveillance. Regulatory T cells, we don't have as much information on these as we do on some of the others, but they are certainly, there is evidence that they are out there. They dampen the immune response either by direct contact with the target or secreting inhibitory cytokines like interleukin 10, transforming growth factor beta. They're important for preventing immune reactions. They suppress self-reactive lymphocytes in the periphery if they should escape the education in the thymus. And there is research into using them to induce tolerance so that transplanted tissues can survive without immune suppression, as much immune suppression. So we got B cells, which become plasma cells, T cells that can become helper T cells, cytotoxic T cells, or regulatory T cells. Whether it's T cells or B cells, they all produce memory cells once they are activated. They require antigen-presenting cells to become activated. The molecules we're talking about that were important include antigens, antibodies, perforins, granzyme, complement, and cytokines. This video is a very good video. I don't think it will play on this, but you should look for it online. Microflix Immunology. And then I'm going to stop here and we'll pick up with immune problems. I will tell you that I moved a couple of slides about transplantation to immune problems because ultimately they are immune problems.